As I told you earlier, the molecular basis of inheritance, the sixth chapter is the biggest chapter in the second PUC, and it is also very important for both the CET and NEET examinations. So far we have covered part 1, and in the part 2 let me start with the DNA duplication. So it was in the 1953 itself, Watson and Crick proposed the model of DNA. While proposing the model of DNA, Watson and Crick, they quoted that it has not escaped from our notice the gene copying mechanism of DNA while deciphering the structure of DNA. Wonderful sentence. It's a wonderful sentence. So while deciphering the model of DNA, structure of DNA, it has not escaped from our notice the gene copying mechanism of DNA. So they themselves in 1953 itself, they showed that DNA copying mechanism takes place. That is very important sentence in the replication. And the replication of DNA is said to be semi-conservative. That means one of the old stand is considered, that is called semi-conservative replication. Semi-conservative replication first noticed by Matthew Michelson and Franklin Stahl in 1958 by conducting experiment. And they took the Escherichia coli for these experiments. And they placed Escherichia coli in NH4Cl N15, that is heavy nitrogen, but it is not radioactivated, and this is N15 NH4Cl heavy nitrogen. When Escherichia coli placed in NH4Cl N15 heavy nitrogen, after some time they shifted this Escherichia coli onto the NH4Cl N14, that is light nitrogen. One is heavy nitrogen, that is N15, and here the N14. They are not the radio activated. So we know everything. Bacteria divides for every 20 minutes. When Escherichia coli shifted from that is NH4Cl N15, these nucleic acids, they absorb this N15 and they have incorporated the N15. After this, when they shifted to the Escherichia coli, shifted to the solution containing N14 NH4Cl. After 20 minutes, as the bacterial cell divides, so the newly formed DNA will show one strand heavy nitrogen and another strand light nitrogen, N14 and N15. Thus, the two new DNA molecules are formed. They are the hybrids after 20 minutes. After again 40 minutes, once again undergo replication, and the results obtained in the ratio of 50-50. So two DNA molecules with light nitrogen N15 and two again the hybrids only. And then after 60 minutes, the results obtained in the ratio of 75 with the light and only the 75% with the light N14 and the 25 hybrids. Hybrids number remains the same. This shows that the newly formed strand and always the light N14 and the old strand, half of the old strand is conserved. Hence the replication is said to be the semi-conservative replication, as noticed by Matthew Michelson and Franklin Stahl. So after this replication of DNA, how the replication of DNA takes place in eukaryotic cell? In eukaryotic cell, as we know very well, 6.6 into 10 to the power of 9 number of base pairs are present. So it requires 38 minutes for its complete replication, whereas only 18 minutes are required in case of Escherichia coli, where the number of base pairs are 4.6 into 10 to the power of 6, and in eukaryotic cell about 2000 base pairs per second replicated. This kind of replication, it requires some energy. This energy can be obtained from deoxyribonucleoside triphosphates, so far not given. Energy required for the replication of DNA comes from deoxyribonucleoside triphosphates, and any mutation that alters the complete replication of DNA. So one method called fourth method of replication of DNA given in the textbook, that is, so the region of ori, that is origin of replication, begins at one end, and then towards one end continuous strand is given off from 5' end to 3' end. This is also very important, from 5' end to 3' end, called leading strand. Opposite to this, discontinuous strands called Okazaki fragments are given off. So these Okazaki fragments are given off from 5' end to 3' end, 5' end to 3' end, like this. So towards one end continuous strand, and towards the opposite end from 5' end to 3' end, 5' end to 3' end, and later these Okazaki fragments are joined by the ligase enzyme. Thus, from single DNA molecule, two new DNA molecules are formed, and in the newly formed DNA molecule, one of the old strand is concerned, that is about the replication. And coming to the important aspect, genetic code. This is very important. Information required for protein synthesis. What do you mean by protein synthesis? Assemblage of amino acids. Assemblage of amino acids called protein synthesis. Information required for protein synthesis is stored in the DNA, and transmitted in the form of code through mRNA called genetic code, which is called central dogma of molecular biology, as I told you in the previous chapter, that was proposed by Francis Crick. Central dogma of molecular biology, that is the DNA giving rise to RNA gives rise to proteins, and the reverse central dogma can be seen in HIV virus, where RNA gives rise to DNA. Reverse central dogma can be noticed in some viruses like HIV, human immunodeficiency virus. So here, the scientists, they wanted to know it. So if the DNA codes for the proteins, that is assemblage of amino acid, that is information required for protein synthesis, stored in DNA, transmitted in the form of code through mRNA. That is in the form of code called genetic code, which is called central dogma of molecular biology. A singlet, for example. So it was George Gamow. He argued that only 4 types of nitrogen base pairs are present at GC, ATGC, 4 nucleotides are present. If 4 nucleotides code for the amino acids, they can code for only 4 amino acids. ATGC can code for only 4 amino acids. A doublet can code for 4 into 4, 16 amino acids. But more than 20 amino acids take part in protein synthesis. Hence, this requires a triplet. So this kind of triplet only can code for 64 amino acids. Hence, it is so George Gamow argued that for coding the 20 amino acids, triplets are required. Only a triplet can code for all the non-20 amino acids which take part in protein synthesis. That is very important. That is the permutation combination of 4 into 4 into 4, that is 64. Later, different types of RNAs are synthesized by Hari Govinda Khorana. And the other contribution of Marshall-Nirenberg is cell-free protein synthesis, not as so far, cell-free protein synthesis by Marshall-Nirenberg. And Severo-Wakao, that is polynucleotide phosphorylases enzymes, that is the contribution of the Severo-Wakao. So because of this, the genetic code or the protein synthesis, because of their contributions, the genetic code or protein synthesis came into existence. And this kind of genetic code or protein synthesis takes place in two steps: transcription and translation. Transcription in prokaryotic organisms and transcription in eukaryotic organisms. First, transcription in prokaryotic organisms. In prokaryotic organisms, there is no nuclear membrane, and this transcription as well as translation takes place in the cytoplasm itself. And here, the transcriptional kit in the prokaryotic organisms, it consists of three important genes, that is the promoter gene and structural gene and terminator gene. Promoter gene and the terminator gene flanks the structural gene. Structural gene is flanked by the promoter gene and the terminator gene, structural gene in the middle. And this promoter gene consists of DNA dependent RNA polymerase. DNA dependent RNA polymerase. This is present in the promoter gene. Promoter gene, it is a promoter gene which releases a DNA dependent RNA polymerase. And the upstream, that is before the commencement, it begins from the 5' end upstream. So this promoter gene releases this DNA dependent RNA polymerases, and it moves towards the structural gene. It starts from structural gene and moves towards the terminator gene. That is from 5' end to 3' end. As it moves from 5' end to 3' end upstream, then from 3' end to 5' end, new strand is formed, that is called mRNA. And the structural gene consists of template and coding strands. Template strand and coding strand. Here, which is very important, it is not the coding strand that codes for mRNA, it is the template strand. This is very important. Template strand codes for the mRNA, and it begins from 3' end to 5' end, and the sigma factor play an important role in initiation of messenger RNA, and for the termination it is the rho factor at the terminator gene present and help in the termination of the mRNA. The newly formed mRNA falls off. So this is about the transcription in the prokaryotic organisms. Whereas in eukaryotic organisms, it is highly intriguing, surprising how only one enzyme, DNA dependent RNA polymerase, involved in the production of all the RNAs like mRNA, tRNA, rRNA. So, but here in eukaryotic organisms, it requires three types of RNA polymerases. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3 are the three different types of RNA polymerases are involved in transcription, that is the production of mRNA, tRNA, rRNA, that is in case of eukaryotic organisms. And here the RNA polymerase 1, it transcribes, that is information will be passed on from DNA to transcribes the RRNAs of different types like 18S type rRNA, and then the 28S type rRNAs, and 5.8S type of rRNAs, and one more type, 23S type of rRNA is also produced by, synthesized by the RNA polymerase 1. It is the RNA polymerase 3 synthesizes tRNAs of 5S type and snRNAs called small nuclear RNAs, and it is the RNA polymerase II synthesizes mRNA. Prior to the synthesis of mRNA, it synthesizes HNRNA, heterogeneous nuclear RNA. This HN, heterogeneous nuclear RNA, undergoes the 3 steps, that is splicing, capping, tailing, undergoes this. Then the HNRNA becomes mRNA. It involves 3 steps, that is in eukaryotic organisms, not in the prokaryotic organisms. So only these 3 enzymes involved, polymerases involved, RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, to synthesis heterogeneous nuclear RNA, HNRNA after splicing, capping and tailing becomes mRNA. So this is how transcription takes place. What do you mean by splicing? So here again the differences between the mRNA of the eukaryotic and prokaryotic organisms. In prokaryotic organisms, polycystonic mRNA, cystine means that portion of DNA which codes for a single polypeptide chain, and 3 different types of mRNAs joined together, they form the polycystonic mRNA that you will study later on in the Lacoperon concept. Polycystonic mRNA. But whereas in eukaryotic organisms only monocystonic mRNAs are produced. Here polycystonic in prokaryotic and monocystonic mRNA. And here no split genes. Here split genes are present in eukaryotic organisms. That means one code word NICE. Non-coding genes are called introns and coding genes are called exons. So introns and exons are present. Exons are present. NICE. Non-coding introns, coding exons. And the non-coding introns are removed. They will be taken out. That is called splicing. Non-coding introns, removal of non-coding introns, that is called splicing in HNRNA. After splicing, capping, adding of, joining of methyl guanosine triphosphate at the 5' end, and adding up of adenylate residues, about 200 to 300 adenylate residues at the 3' end, called capping. Because of this splicing, capping and tailing, that is adding of 200 to 300 adenylate residues, that is called tailing. Because of this thing, HNRNA becomes mRNA. So that is about transcription. And coming to the translation, the language of genes will be translated into language of proteins. It involves all the RNAs, mRNA, tRNA, rRNA, etc. They take part in translation. Language of genes will be translated into the language of proteins. The one important thing in translation is, as amino acids do not have structural organization to recognize codon on mRNA, that is triplet present on mRNA, as they do not have the structural organization. This is very important. So far not asked. Amino acids as such do not have structural organization to recognize the codon on mRNA. It is the anticodon of tRNA recognizes the codon and brings a particular amino acid. Does it act as an adapter molecule, which is very, very important for your NEET examination. As the amino acids do not have structural organization to know about the codon on mRNA, it is the anticodon of tRNA recognizes the codon, and the tRNA brings a particular amino acid onto the mRNA. That is, hence it is called transfer, and it acts as an adapter molecule. tRNA resembles clover leaf in its model, consisting of 4 ends, 2 ends, 1 end. Anticodon, opposite to this, there is site for the attachment of amino acids. And the language of genes converted into language of proteins during translation. It involves an enzyme called amino acid synthetase and ribozyme enzyme. These are the 2 important enzymes. So this amino acid synthetase, this initiates the translation, and the translation takes place in 3 steps: initiation, elongation and termination. Genetic code always begins with AUG. AUG codes for methionine and genetic code. So the details are studied in only the prokaryotic organism. There the ribosomes of 70S type present, the subunits of 70S type, 30S type and 50S type. As soon as mRNA mRNA formed, and along with the ribosomes it forms a polysome or polyribosome. Genetic code begins with the AUG. It is the amino acid synthesis which initiates, which activates the tRNA and facilitates the attachment of amino acids after knowing the codon. So the AUG, genetic code begins with the AUG, and the anticodon having UAC brings a particular methionine, that is tRNA activated by amino acid, and facilitates the attachment of methionine at the site of amino acid attachment, and this is called initiation. And then it brings a particular amino acid, and the ribosomes consists of more than 80 different types of proteins which play an important role in protein synthesis, and the rRNA of 23S type rRNA, and an enzyme called ribosome. This ribosome enzyme, not made up of proteins, non-proteinaceous enzyme, play an important role in this elongation or in protein synthesis. So in the textbook it is given, that is AAU codon and UUA, that codes for aspargine. So 20 different types of amino acids, for 20 different types of amino acids, 20 different types of tRNA present, and as the tRNA recognizes the codon AAU, it brings anticodon having UUA brings aspargine. If the codon is GCA, then the anticodon CGU that brings alanine. That brings alanine. That is also very important. If the anticodon UAC, if the anticodon AUG, if the codon is UAC, that brings tyrosine. These are some examples given. If the codon AGU, then it is UCA, that brings serine. So all these different things given in the book. What are all the given about aspargine, alanine, and then the next one is serine and tyrosine, I explained, and then it is the ribosome and the 23S RNA helps in the binding of one amino acid with the another one. mRNA moves through the ribosomes, and anticodon of another tRNA recognizes codon, brings another amino acid, binds to the first amino acid and floats back into the cytoplasm. Hence it is called soluble RNA. Then mRNA moves to the ribosome, anticodon of another RNA. This will be continued till the arrival of nonsense codons. Nonsense codons are UAA, UAG, UGA. Once the UAA, UAG, UGA, that is in sequence. Once the nonsense codons appear, polypeptide chain will be terminated. Thus the protein synthesis takes place, and this is called UTR, untranslated regions. And the release factor also helps in the termination. This is about the translation in detail. And the last one is characteristic features of genetic code. One code must does anta. Genetic code is meaningful, unambiguous, and specific. Must anta one code. Meaningful, unambiguous, very specific. That means in all organisms, AUG codes for methionine, whether it is a prokaryotic organisms or in that is universal, meaningful. So that means here. So the codon which codes for one amino acid, particular amino acid, will not code for another amino acid. So this is very important. The codon which codes for a particular amino acid, for example, AUG codes for only methionine, not another amino acid. This is thus the genetic code is meaningful, unambiguous, specific. That is must, and it does. That means an amino acid is coded by more than one codon. More than one codon. For example, glycine not only coded by GGG but also GGC, GGA, GGU, and alanine not only coded by GCC but also GCA, GCU, etc. And the valine not only coded by GUU but also GUG, GUA, like this. And the arginine not only coded by CGG but also CGC, CGA, CGU, and proline not only coded by CCC but also CCA, CCG, CCU, and the leucine not only coded by CUU but also CUG, CUC, CUA. So like this, that means amino acid is coded by more than one codon. That is degeneracy. But the codon which codes for a particular amino acid will not code for another. That is called meaningful in the beginning. So here, but the amino acid is coded by many codons, but the codon code for a particular amino acid. So this is called degeneracy of codon. And genetic code is universal. As I was telling you, AG codes for methionine. It is common in whether it is in prokaryotic or this is called genetic code is universal. However, there are exceptions in the DNA of few protozoans and mitochondria. Exception is with Kedilla, the genetic code is universal. And there are starting codons and terminating codons. Starting codon is AUG, terminating codons are UAA, UAG, UGA. And particular order UAA, UAG, UGA. ಅದಕ್ಕೆ ನಾವು ಇದು ಕೇಳ್ತಾರೆ ನೈಟ್ರೋಜನ್ ಬೇಸಿಸ್ ಆನ್ DNA UAA ಅಂತ ಹೇಳಿದ್ರೆ ಅಲ್ಲಿಗೆ UAA ATT ಅಂತ ಬರುತ್ತೆ. ಅಲ್ಲಿ ಥೈಮಿನ್ is present in DNA. And then UAG ATC ಅಂತ ಬರುತ್ತೆ. UAA UAG and UGA ACT ಅಂತ ಬರುತ್ತೆ. ಸೊ ಇದನ್ನು ಕೂಡ ಕೇಳ್ತಾನೆ, ಹುಷಾರಾಗಿ ನಾವು ಇದನ್ನೆಲ್ಲ ನೋಡ್ಕೋಬೇಕು. ಆಮೇಲೆ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಆಗುವಾಗ ಯಾವ ಥರ ಆಗುತ್ತೆ ಅವೆಲ್ಲ ತಿಳ್ಕೊಳ್ಳೋವಂಥ ಅವಶ್ಯಕತೆ ಇರುತ್ತೆ. ಈಗಾಗಲೇ ಹೇಳಿದ್ದೀನಿ, ಬೈ ದ ಅಡಿಷನ್ ಆರ್ ಇನ್ಸರ್ಷನ್ ಆಫ್ DNA, pairs of genes of DNA, this causes frame shift mutation ಅಂತ. ಫ್ರೇಮ್ ಶಿಫ್ಟ್ ಮ್ಯುಟೇಷನ್ ಅಂತ ಅದು ಸೀಕ್ವೆನ್ಸ್ ಎಲ್ಲ ಕರೆಕ್ಟ್ ಆಗಿರಬೇಕು. ಅದು ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಆಗುವಾಗ ಅಲ್ಲಿ ಇನ್ನೊಂದು ಪಾಯಿಂಟ್ ಏನು ನಾನು ಮರ್ತಿದ್ದೆ ಅಂತ ಹೇಳಿದ್ರೆ, ಒಂದು the template strand gives rise to different types of RNAs. If both the strands give rise, means it results in double stranded RNA that silences the mRNA. Double stranded RNA translation will not takes place. ಅದಕ್ಕೋಸ್ಕರ ಒಂದು template strand it codes for it is responsible for the mRNA production ಅನ್ನೋದು. ಸೊ ದಿಸ್ ಆರ್ ಆಲ್ ದ ಥಿಂಗ್ಸ್ ಆರ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ತುಂಬ ದೊಡ್ಡ ಚಾಪ್ಟರ್ ಆಗಿರೋದ್ರಿಂದ ಮತ್ತು ಇವೆಲ್ಲ AGU AGC ಈ ಥರದ್ದೆಲ್ಲ ಓದ್ಕೊಳ್ಳೋದ್ರಿಂದ ಒಂದು ಸ್ವಲ್ಪ ಹುಷಾರಾಗಿ ಇದನ್ನೆಲ್ಲ ಓದ್ಕೋಬೇಕು. ಏನೆಲ್ಲ ಅರ್ಥ ಮಾಡಿಸ್ಬೇಕಾಗಿದ್ದು ಅದು ಅರ್ಥ ಮಾಡಿಸಿದ್ದೀನಿ. ಇದು ಪಾರ್ಟ್ 2 ಮಾತ್ರ. ಪಾರ್ಟ್ 3 ಇದು ಬಿಗ್ಗೆಸ್ಟ್ ಲೆಸನ್ ಆಗಿರೋದ್ರಿಂದ ಪಾರ್ಟ್ 3 ಇರುತ್ತೆ. ಪ್ಲೀಸ್ ಸಬ್ಸ್ಕ್ರೈಬ್ ಆಲ್ ಆಫ್ ಯು, ಆ್ಯಂಡ್ ಮೇ ಗಾಡ್ ಬ್ಲೆಸ್ ಯು. ಥ್ಯಾಂಕ್ ಯು.